Oh, hello there. Gosh, I was really trying to get away from recording things because this is how you guys have learned it like the first way. But there was just too many students that had things going on after school or being lunch, and they don't want anyone to feel like they're at a disadvantage for not being able to come after school in person. I get that. So if you're able to come, or if you were there, great. This is just for you. But actually, this is good for maybe next year when you go to take the test. You'll have these kind of like big reviews. Okay, so just a warning. I'm going to like skim over the entire topics and really just talk about the big main idea so that you have this like kind of framework. This should be review, right? So we shouldn't be learning things from this. We should be like, oh yeah, that's right. Oh right, that's good to know type of thing. So here's what I want you to do if you're at home, and I'll say this in person. What I want you to do is like get out your notes, like start with topic one. And as I go through that review, like then pause it afterwards and like kind of scan back through and go through it, right? So we're just kind of refreshing our brain to where it used to be, right? So that's what you should be doing at home. Okay. All right, friends, let's start with topic one. Or not, because it's not going forward. There we go. I'm going to show you the big idea for each topic. So you kind of like, why are we talking about this, right? So topic one was our big idea is like, how is life the same? What are the unifying characteristics of life, right? And then we'll talk about how it's different. This also kind of outlines our entire year. So how are all cells the same? We started with talking about the functions of life, those characteristics of life, right? This was our acronym as Mr. Shang. By this point, we have like 10 acronyms of ways to remember things. So you need to start incorporating these acronyms into like a sentence that gives it more context, right? Or context, right? Like Mr. Shang and his functions of life, right? So like add context to it so that you just don't have all these PRAD, blah, 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 kind of flying around, right? So we talked about that. These are the 7 basic life functions. And if you look through those, we're basically going over them for the entire year, right? Growth, we're talking about that in mitosis. Nutrition, exchanging between cells. We're going to be looking at how cells get rid of things, how they maintain internal environment. So know these things, right? Go through them. Remember Mr. Shang, test yourself on that, and then some kind of example of what that means. Be able to list and give an example. Okay, so these are all the 7 functions that make cells the same, make life the same. The other part is where the cell theory. We've gone through this a couple different times. You were tested in already, like the 3 basic parts. And then we kind of looked at the things that go against it in 1.5, right? All cells come from pre-existing cells. Cell is the smallest unit of life. And what the heck? All cells can come from pre-existing cells. I know I forgot what I said. Cells from pre-existing cells, cells like that. Oh, all living things are made of cells. Gosh, Mrs. Myers. Okay, I need a review. All right, but the thing I did want to put on there for you to kind of refresh your brain around, because you have all that in your notes. Did you put the exceptions in your notes? Because there was 3 specific things and mentioned in lecture and your book and in IB. Like, so these are our examples, just know them. So the striated muscle, the giant algae, and the aseptate fungus, right? So know that what makes these all different is they have like, first of all, they're really big, right? Instead of the small. So they're not exactly going against those 3 parts of the cell theory, but also they have multi-nucleated, multiple nucleuses. Tanisha and I were talking about this during first period, right? We looked it all up. That is what makes them different, is they have like, well, first of all, these muscle legs are really long. The giant algae is giant, right? So really big. And then this continuous cytoplasm. But what makes them all very different is that they have multiple nucleuses in each cell, nuclei. Okay, so know that that's kind of that key difference. Okay. Other thing we went over in topic 1 was just magnification and the surface area versus volume. This was a big issue. I think everyone I talked to today had some kind of question about, so that I went through that with a lot of you. Make sure you go over that again. And the magnification, that's kind of stuff that we just need to practice and know, right? Some of those things that that you should have in your notes, that you should definitely be able to use, right, is our MIA. Remember that when I'm going to do my calculations, whichever one I'm trying to calculate, I cover up, right? And then so magnification is image divided by actual. Image is magnification times actual, and my actual is image by my magnification. Remember, the trickiest thing you guys kept getting is this, right? The nanometers, the micrometers and the millimeters. Remember that if you always measure in millimeters, right, then you're just going to multiply that by 1,000. So you're adding 3 zeros to get my micrometers. Okay. Other kind of key point, magnification has no units. Do not put units on there. For image and actual, make sure we put our units. All right. Oh my gosh, give me that. All right, surface area to volume. Remember that we're not looking at as one thing increases the other. We're looking at the ratio, the comparing of one thing to another, right? So we talked about when I have just a simple single like cube, right? So it's like 1 by 1 by 1 by 1 by, right? Like then I'm looking at like the surface area, if I add it all up, is 6 and the volume is 1. So I've got like a 6-to-1 ratio. However, when I'm looking at a lot of different cubes put together, all that surface area in the middle is not exposed. We talked about like it's like a towel. If I drop a towel into the pool and I take it out and I leave it all bundled up, it's never going to dry because there's not the surface area on the outside. However, if I lay it down flat on the ground, tons of surface area, right? So it's going to dry really fast. Exchange. Cells, tissues are the same. They want to increase their surface area to optimize transfer. So that ratio, as it gets bigger, goes down, and that's not good, right? So my surface area to ratio at the smallest size is going to be the highest. And as it gets bigger, as my cell gets bigger this way, right, with the volume and the surface area, that ratio goes down. So make sure that we know those things. If those are in our notes, again, lots of people had questions on that. All right, pause, go through your 1.1 notes. Okay, 1.2. How are cells different? So from 1.1, we talked about how are they all the same? Now we're like, okay, but why are they different? All right. We put so much effort and energy into this. I need to make sure you guys go through your things that you have. I am not going to talk about all this, but here is all of the things you did in all those labs, right? First of all, Dora and her prokaryotic versus eukaryotic. You need to know the difference between those two. You have drawn them, you've listed them, you've been tested on them. Make sure you just go back and review those things. How their DNA is structured, what organelles do they have, how they replicate. We just went through mitosis, so now you know a bunch of that. Binary fission, right? Those two things, and then their average size, right? So smaller and larger. Know the structures, know how to draw them. These are the very basic kind of functions of those cells. Of those organelles. So make sure, there we go, that I'm able to draw, label, explain, list, right? This is the fun stuff. You know this. Just kind of go back through and refresh your brain and use what you did, like those activities, like the day without a cell. Okay, what happened if we didn't have ribosomes? What happens if we don't have a nucleus? What does the Golgi do? Now that we've gone through a little bit more of knowing that like DNA to the endoplasmic reticulum and then the Golgi like export it, and now we've talked about endocytosis, hopefully that stuff's starting to kind of, come together. You're like, no. Okay, anyway, right? Or that the nucleus and the centromeres, we just talked about, the microtubules in mitosis, you're going to see how these things start to kind of build on each other. The vesicles, vehicles that allow, that temporary storage for exocytosis and endocytosis. All right, so go through those. Those are in your cell organelle lab review. Draw, list, explain, compare. Okay, remember in our drawings here, just a couple, like when we do that flat, make sure, actually, if I go back, wait, can I go back? Remember in our prokaryotic that the nucleoid contains like all that DNA, but there should be no line around it because that means that it's a membrane, right? Versus in my animal cells, my lines can be completely together, right? So vacuoles size. A lot of you are really nervous about getting the micrographs and being able to identify. Just make sure you look at all the ones you've seen because I tried to give you all those examples that I've seen. Okay, this isn't the best example here because I don't like that this is all the way around. But remember that your lines should be drawn to the outside, right? And that should be very clear where it's going and what it's pointing to. All right, remember that our plant cells are this way, right? They're more square. We looked at those in mitosis and that it has that cell wall on the outside in the chloroplast. Those are my differences. Okay, 1.3, so pause, go through all of that. Right? 1.3 and 1.4, big ideas. This is all about membranes, right? So what are membranes? How are they built? What the heck do they do, right? Like how do they move things in and out? This was like a big unit, especially with like the history of how things that, like you just need to know a lot of the nitty gritty. However, when we went through like transport, moving things back and forth, hopefully it's all starting to come together, right? But think about these big questions, right? Like why, if I don't water a plant, does it start to wilt over? Why can't I drink seawater if I'm thirsty? Why does that kill us, right? Think about that. When I'm drinking a bunch of salt water and because I'm thirsty and my cells are just pushing it back out, right? Then I'm going to dehydrate myself, right? Why do sugary drinks dehydrate me? What happens when I put like cells, if I'm not enough salt versus too much salt? So big idea, and then we'll be going to the small levels. Okay, just some basics about how it's built. You guys have drawn these, you've listed them, you had that test question. Most of you did really well with drawing the membrane. I went over in class today, make sure that you use words like hydrophilic and hydrophobic. Do not use attracted and repellent. You can say that what that means, but make sure you use those keywords, right? That the hydrophilic and hydrophobic. If you use words like it's up on the board, like water loving or water hating, no points, even though we know what you mean. Okay. Remember that one of these is called a phospholipid. Put together, they're called the bilayer, right? That's a big thing. And then you should know that it forms in that opposite because you get the hydrophobics coming together and the hydrophilics, the water loving, are the outside and the inside. There's a word missing here. So that we have that. Okay. The models. Remember that you need to know the Davison-Donnelly and then how it went to the Singer-Nicholson and what was wrong with this model or how it was different from here. You guys drew this out. You've had test questions on it. Again, go through that last test that you've already gone through, right? So they were saying the 2 layers of protein were on the outside and that it was kind of like a sandwich with the proteins on the inside. Big idea, Singer-Nicholson came along and they said, there's actually these proteins that are embedded. So they had them on the outside. Remember your test of little squiggles. Singer-Nicholson came along and did these embedded proteins, right? The transmembrane embedded proteins. So make sure you know the difference between those, like how are you going to remember that? And that there was that kind of transition in models, okay? All right. Whew, there's so much here, right? So I kind of put them all together and there's nowhere that my face is going to fit. Here we go. Let me put on the rat. There you go. You can just laugh there. Okay. Functions of membrane proteins, the fluid mosaic model. So being able to draw that, you've already done it. And then the types of transport. So much. But again, big idea. Why do I need proteins in the membrane? What the heck do they do? Transport, right? We learned about this, right? And then we're going to look at these things too, right? They form those tight junctions, so they put them together. Enzymes we'll talk about in proteins, but this transport passive active, they have those anchorage sites. Where are the anchorage sites? Are they in here? They're in the other one, where it shows that it attaches, so the peripheral things can attach. And then transduction, just for now, know that they have those membrane proteins. You can know the jet rat, but be able to go through the things that we've talked about, right? So we've talked about junctions, we've talked about transport, and a little bit of anchorage and attachment sites. Focus on those things, okay? Know that there's recognition proteins and enzymes, but don't need to get into the nitty-gritty of that. This was my fluid mosaic model. Remember, the mosaic is the piece of pieces, but fluid meaning that things can go in and out. 2 different types of transport. Start with passive, one that does not require, I look really funny on top of this wrap. Okay. One that does not require energy and the one that does, right? Remember that in your test, you need to mention the word concentration. You can't just say high to low. It needs to go from areas of high concentration to low concentration. Okay. So passive, no energy required. I have simple, facilitated, and osmosis. So if you have to outline passive transport and examples, simple diffusion, facilitated diffusion, osmosis. We looked at osmosis in a lab. You should be well aware of that one. And facilitated diffusion, we talked a little bit about with the phosphates membranes, going back to the voltage, right? So no energy in either of these, right? But so this is water. So osmosis is just water. Facilitated diffusion is when I need some kind of help getting across, right? So it's 2 cannot freely cross the membrane. They're either charged, or they're bigger macromolecules. And so I've got membrane proteins to help out. Still no energy required, but it does require these different channels. This one's just open. This is a carrier. The membrane opens and closes. So proteins have those hydrophilic pores. They let it through. Carriers actually change the way that they have a passage or versus something that carries across. Okay, the other type, really quick, my different types of solutions. Hyper, iso, and hypo. Yeah. So I was talking with you guys a lot today in class. Always start with the end result. So if you always start with the end result, it's so much easier to kind of go through, right? So if I have a cell that shrinks, right? Or if I say, I'm sorry, if I remember in hyper, like people are super hyper, right? Get really small, right? Burn off all their energy. Now they're tiny, right? Hypo means below, right? So my hypo is going to swell. So if I say like, what happens to a cell in a hypertonic solution? Okay, hyper is going to be small. So why is it small? Well, it's small because it lost water, right? So the water has to move out. Well, why would it lose water? There has to be some type of difference. So if it's losing water, that means water is going this way. Oh, that's trying to dilute the solution around it, right? So there must be more molecules around it than there is in it, and I'm trying to move water. Start the other way. Hypo. Okay, I remember hypo, hippo, it swells. Okay, all right. So why would a cell swell? Well, it would swell because water is moving in, right? Why would water move in? It would move in because there's more inside, right? Solute or sugar, salt, whatever you're talking about, than outside, right? So you can work through those things and then figure out where is it moving, why is it moving, okay? The other type of, and we'll look at the lab and the data and the things like that. The other type of transport that we talked about is active. And so this is active right here. This is still osmosis. We're looking at passive. This is active over here, right? Which means it's a big bulk transport, right? And looking at cytosis. So I'm going to show you that on the next one. Hold on, let me clear back here. Okay. Oh no, I don't know. I must have forgot endo and exocytosis. All right. And then I don't have the transport. Okay, I forgot a slide here. I will go back and add it. Well, actually, hold on. Okay, magically added these. Okay, just forgot active. Sorry, very important. So active transport, right, is requiring energy because I'm going low to high. Remember, to get my grades to go from low to high, I need to put some energy into it. You need to know those examples. The biggest one's talking about bulk transport with cytosis. So endo, into, exo, exiting, and my protein pump. The tricky one we are looking at today in class. So the big example of active transport is your sodium potassium pump. But potassium, if it just says potassium ions, that is going to be with my facilitated because they're charged. So if it's just potassium, right, that one kind of triggers some people. We're looking at that charged, and that's just going through facilitated. If it's sodium, sodium potassium, that is pump, right? You can think of like energy into a pump, okay? And then the reasons behind the sodium potassium pump, I told you guys today in class. You will go over so much of that when we talk about neurons in topic 6. So for now, just know that it's dealing with going from low to high sodium, and that it requires energy. These ones right here, remembering that the direct is the one where that energy is used to mediate, and this indirect one is just kind of coupled with it, so it uses it. Right? So active, no, the pump, and then cytosis. Okay, pause, review. 1.5. Okay, 1.5 big ideas, chicken and egg. There you go, summary. No, sequence of life formation in our evidence, right? So how, if all living things come from pre-existing cells, then where the heck did the first one come from? This is our theory of how life came to be with all these different pieces and what some scientists did to try and prove it, okay? Or kind of recreate it. All right, remember that our cell formation starts with non-living inorganic compounds, not carbon. We've got our nitrogens, right? We've got these really simple, someone asked today, what's a monomer? Remember that monomer, it means one, right? Remember someone who has mono, just by themselves. We're able to change into polymers, which is many, right? Think of like polygon. So they assembled and came together, and then they became capable of replicating themselves and gave rise to our first like protobiotes, the simple cells, right? They are creating like this. So they're like, okay, that's cool. How the heck would that even happen, right? So if you're thinking that it's going from like hydrogens and nitrogens and some oxygens, like, and they just like came together. What? Like, okay, so Miller-Urey, they were trying to recreate, well, how would that happen, right? In their experiment, they boiled the water, created the vapor, added some kind of electricity, and when it cooled, they found that that type of process made organic molecules. So if ancient Earth, volatile, all these different things in the atmosphere, lots of electricity, right, going around. And then that's how these more complex polymers were able to form. Okay, so that was the experiment that showed that I can get mono to poly, that I can get really basic things coming together to make a complex thing in the presence of electricity. Okay, all right. Oops, sorry. Clear. Nope, mouse. Okay, all right. The other parts, okay, well then how did that lead to life, right? And so our big kind of thing was talking about how the cells went from what you need to know is like basic elements, monomers, polymers, came together to make the first cell. And so that process was called biogenesis. What Louis Pasteur was trying to, we've gone over this so many times, but what he was trying to show is that like biogenesis right now cannot happen under current conditions. So the earth we have now, we don't just get spontaneous growth of new cells, right? And so his experiment was trying to show that cells can only be formed by pre-existing cells, right? He was trying to show that we don't have these anymore, but that can come from that living cells living cannot arise independently by a biogenesis. So biogenesis was first. He was trying to show that those conditions don't exist anymore. Remember, the conditions that did exist in the past were thermal vents, volcanic, right? Looking at those things that happened when Earth was forming, and now. This is getting long. Okay, so remember, he boiled the broth with the microorganisms. They were alive. He boiled them to kill them. But we have these little microbes in the air. And so if I heat them and it's actually sealed, I get no growth, right? So if I have this sterile this place where it's no exposure to the air where I have microbes, I get no growth. If I am exposed, I get microorganisms into the broth. I get contaminations. They start to break it down. Okay, this was shown differently on your practice question, so I really want you guys to look at that one, where it's like the X, Y, Z, like which one actually grits the microscopic. Oh, I don't know. I don't have time for a dentist appointment. Oh no. And looking at how that was broken. Okay, so Louis Pasteur was trying to show that biogenesis does not happen on current Earth, right? It cannot arise, right? That only old situations like with like thermal vents and hydrovents and all that, that's where we would get life now. Okay. Know the RNA world hypothesis, that's where like it poses that it was the initial chemical instruction. You can read through that, but just know that that's where it was thought. And our evidence for that comes from that I see that those two, remember these two, oh my gosh, I can't, there we go. That those two different molecules, it's not letting me draw anymore. I'm sorry. So mitochondria and chloroplast, that is our evidence because they have their own membrane. They can divide like vision-like, like prokaryotic. They have their own DNA, and the ribosomes are more like, what the heck? I'm sorry. Our ribosomes are more like the prokaryotes and the eukaryotes, right? So they are believed to have evolved from endosymbiosis, and the number of features in the organelles is what proves that. Okay, all right, here we go. Oxygen enrichment, knowing like how oxygen changed over time, right? From going in the oceans to going to the atmosphere. Just know that, like, generally speaking, it reacted with the iron in the water, and all that iron came out and dissolved and created those pores. Those iron-rich deposits. That once the iron was out, because that was highly reactive, oxygen could go into the atmosphere. Once it was in the atmosphere, it could create the ozone layer. So I see these changes of life from water to land, and the changes of that are because of the oxygen changing. Okay. 1.6, last one, big idea. How do cells grow and divide? Why do they divide? And what happens if it goes wrong? How, why, and what happens if it goes wrong? Okay, big idea. There's a cell cycle. 75% of the time is spent preparing to divide, and then the actual 25% is actually dividing. There are specific phases that it goes through, just like packing things up and moving away. You need to know that G stands for growth. So there's two areas of growth. The first one, knowing what happens there. The second one, knowing what happens there. And then in between, the DNA replicates. So, okay, I'm going to grow a little bit, get some ready, increase my metabolism. Now I'm going to double my DNA, and now I'm going to make sure I have all that stuff ready to go before I actually split in two. All right? This M phase, 25%, is where my different stages come from. We'll talk about in a second. So this first one, enter, remember it's entering to the cell cycle. It's like an interim. It's kind of like the pause in between. I get all of these things happening. Just imagining, what do I need to divide a cell into two and have the identical? Well, I'm going to need double DNA. I'm going to need double organelles. I'm going to need to grow. I'm going to make sure that I have all the proteins, I have all the nutrients, and that I have the energy, right? So if I have all those things, it's going to be able to divide to do and create two separate things. So knowing that the interphase, that's the intentions of it, is to get it ready, and the M phase is actually for dividing the DNA and splitting. Mitosis, you have those stages. You can kind of now start to go through some acronyms, or I want to say mnemonic devices. So PMAT, right? Or you can remember IPMAT, interphase, and then PMAT. So prophase, metaphase, anaphase, telophase. I talked a little bit about some mnemonic devices, right? That I get all my pros together and they're all condensed because they are ready to go, right? Woo, before a game. And then they meet in the middle of the field, right? Metaphase, right here in the middle. Of course, you need to know about like the microtubules and all those things, but big idea, meet in the middle. And then all of this chaos happens, and Anna, she splits them all up, right? And they start to move to opposite sides, and they make new teams. Telophase, we used to say like they call, it kind of looks like a telephone. You guys don't use that anymore. But just know, pros get together, they meet in the middle. Anna divides them, right? She's like, you're on this team, you're on this team, you're on this team. And then they create two new teams in telophase, and it's cytokinesis is the actual one that puts them in half. Calculating mitotic index. Make sure you know how to do that. How many cells are in mitosis? What's my total number of cells? It should tell you. All you have to do is add them together and divide. Okay, cool. All right. How do I regulate that? Okay, cyclins. That's right here. I'll go right here. So know that those checkpoints, that after each checkpoint, it's like stop. How much time, right? Do I have enough? If I don't, the cell cycle will stop, right? So know that G1 checkpoint. Am I prepared? G2 checkpoint. These are things that happen to make sure that things are where they should be, right? And then I've got my M checkpoint. Make sure it's ready to split apart. Cyclins are those regulatory proteins that control the progression of this. So my cyclins are the proteins that control that. I can't draw anymore. So think of this. The complex, like they trigger the cell to the cell cycle. And if it's not there, right, it's not ready, it's not going to keep going. And after it's done, it breaks out and it breaks up and it's inactivated. So know that cyclins regulate the cycle. What happens when it goes wrong? Cancer. You do need to know the difference between a tumor and then my, benign versus, why is that not moving? Sorry, my different types of cancer. So malignant versus benign. Metastasis is the spreading of cancer. What would that look like? I get tumors in other places. So I find breast tissue in the liver, right? Or if I find liver tissue, if I have liver cancer, it originated there. There's different stages. You don't need to know about them right now, but you do need to know the oncogenes, right, that actually create that, the carcinogenics, right, the ones that, the mutagens, the things that can cause. And the big thing with correlation and causation, I've seen that a lot. What you can tell for sure, right, where it's like, what can you conclude? If it asks you to conclude something, watch out, because that's trying to trick you that like, oh, well, I can conclude that if you smoke more cigarettes, that you have a higher incidence of cancer, right? That this causes this. Cigarettes cause cancer. I cannot conclude that. But what I can conclude that as my cigarettes per day increase, the incidence of cancer increases, right? So it could be that people that smoke a lot of cigarettes every day, right? Like maybe are in the sun every day, they're outside. And so maybe that's the thing that's causing the cancer. So you can see a correlation between the two, but it does not mean that cigarettes cause that. So make sure you're just reading the graphs for what they are. Okay. And my friends, that is the end of topic 1. Big review. Make sure you go through your notes, pause in between, and you know this stuff. I believe in you. Believe in yourself.